Is that thing? Is it beefy?
Yeah, I went in today just to like.
Yeah.
Yeah. 90.
Those days.
That's like how skating. I like have to like I don't have
I just work.
Yeah.
At least for one.
whatever. 7 days.
Planting them, that one in your yard means boycotting or something.
I do like this.
So,
Yeah, good catch. Like I watched what's going on?
Like
You could be totally ignorant.
Okay. Hi, everyone. Can everyone hear me? Okay, it sounds like the mic is working. Great. So thank you so much for coming today. I'm really excited to be back. This is kind of my home departments where I did undergrad, and then I worked at the herbarium for a number of years after undergrad. So yeah, thanks for having me, and it's cool to be back. So let me make sure my keyboard is working. All right. So I'm going to start today with just a bit of a background about the types of questions and systems that I like to think about. And then I'm going to talk about 2 projects today. One that I did as part of my dissertation work, and then another that I've been working on during my postdoc with Emily Grman at Eastern Michigan. So my research concerns the symbiosis between leguminous plants and rhizobium bacteria. Rhizobia live in nodules on the plant's roots, where they fix atmospheric nitrogen into forms that plants can use. And the plants, in exchange, reward the rhizobia with carbon that they've fixed through photosynthesis. And it's kind of a lot of carbon because it takes a lot of energy to break that triple bond of nitrogen gas. This can be upwards of like 10–15% of a plant's total carbon expenditures. So it is a pretty expensive symbiosis, but it's often worth it because nitrogen is so often a limiting resource. So I think this symbiosis is just really cool, but it's also really ecologically important because rhizobia are responsible for nearly all non-anthropogenic terrestrial nitrogen fixation. Our other major terrestrial input being synthetic fertilizer. And synthetic fertilizer also takes a ton of energy to make, but now the energy is coming primarily from fossil fuels rather than from sunlight. So if we can understand how this symbiosis really functions, it has the potential to reduce our dependence on fossil fuels. However, our overuse of fertilizer may actually be disrupting this mutualism and thus increasing our dependence on fertilizer. And to explain why that might be happening, I'm going to explain some classic mutualism theory that combines ecology and economics. So mutualisms are simply species interactions where both species benefit. And historically, they were understudied compared to antagonistic relationships like competition or predation. And that's partly because of biased interpretations of previous results, but also because of some of the early theoretical ecology models returned nonsensical results if you parameterize them for mutualisms. Like if you take the Locke-Volterra equations and you put in mutualistic parameters, it gives this completely nonsense result where both species grow to infinity. So mutualism theory compared to things like competition theory came off to kind of a slow start, but of course now it's a very robust field and we have really useful predictive frameworks. For example, resource mutualisms can be modeled as a biological market, so borrowing from economic theory. And here, both species are trading resources with the other and both benefiting from that trade. So here, this diagram is describing the relationship between plants and mycorrhizal fungi. So on the x-axis, we have carbon, and the y-axis is phosphorus. And these axes represent how much of each resource the organism can accumulate in a given year. So R2 max here is the most amount of carbon that that plant could accumulate if it put all of its energy into accumulating carbon and none into accumulating phosphorus. Likewise, R1 max is the maximum amount of phosphorus that plant could possibly accumulate if it didn't try to get any carbon and only worked on getting phosphorus. And so this is assuming a fundamental trade-off between acquiring these 2 different resources. But that is pretty reasonable because if you're a plant, you only have so much energy to go into making tissues, but the kinds of tissues you need for getting carbon are shoots and leaves, but the kinds of tissues you need for acquiring phosphorus are roots. And so if we assume this trade-off and interpolate between R1 max and R2 max, we get what we call the isolation acquisition isocline. And this represents all of the combinations of carbon and phosphorus that this plant could accumulate. But of course, if you're a plant, you need both carbon and phosphorus to grow. And so the best strategy lies along this optimal consumption vector, the 1-to-1 line. So along this vector, the plant is accumulating a balanced amount of each of these resources. And that means that the best strategy is here at this intersection between the isolation acquisition isocline and the optimal consumption vector, where the plant is accumulating as much total resources as possible, but in a balanced way, so it doesn't have a surplus of one or the other. So this is the best that the plant could possibly do when acting alone. It would have 50 units of carbon and 50 of phosphorus. But participating in mutualism gives this plant another option. So here is the trade acquisition isocline. It can't accumulate additional carbon. R2 max doesn't change, but by trading with the mycorrhizae, the plant can accumulate more units of phosphorus per unit of carbon, and so that slope of the isocline is much steeper. And that means if you follow where that isocline intersects with the optimal consumption vector, you can see that by trading with the mycorrhizae, the plant can now accumulate more carbon and more phosphorus during this growing season than it could when acting alone. And if you sum those two gains, you get this vector G, which is the gain from trade. So this is how much the plant is benefiting from participating in this mutualism. And likewise, we can make the exact same figure, but from the perspective of the mycorrhizae. So now we have a fixed R1 max. The maximum amount of phosphorus that this mycorrhizae could accumulate doesn't change by trading with the plant, but now that trade acquisition isocline increases the maximum amount of carbon that the mycorrhizae can accumulate, and again, it gains a benefit from participating in this trade. So since both species here are benefiting from participating in this trade, this is a mutualism because both species are doing better than they would working alone. And so generally, if there's a significant gain from trade for both species, this is a nice, strong, stable mutualism. But environmental conditions can shift these isoclines and change the benefit of trade. So to demonstrate how that works, now I'm going to switch to talking about the legume-rhizobium mutualism, and just from the perspective of the plant. So here, my x-axis is still carbon, but now my y-axis is nitrogen instead of phosphorus. So if we have a plant growing in low nitrogen soil, the maximum amount of nitrogen that it can accumulate, even if it puts all of its resources into accumulating nitrogen, is really low because it would have to grow a really huge root system to glean that little bit of nitrogen out of the soil. And that means that if it instead strayed with rhizobia, it has this really big gain from trade. There's a huge benefit for participating in this mutualism. However, if the plant is instead growing in high nitrogen soil, now that isolation acquisition isocline is higher because it doesn't take as much energy being put into root growth to get that nitrogen out of the soil. And so growing in high nitrogen soil reduces the benefit that the plant gets from trading with the rhizobia. It's still beneficial, but less so. And if the benefit of trading for rhizobia is decreased, the plant may reduce its investment in rhizobia. And then, of course, rhizobia cannot fix nitrogen if it's not given carbon, so it'll reduce its nitrogen fixation. And this has the potential to create a feedback that destabilizes this mutualism. And if it's just a temporary thing, this might just be, a temporary reduction in the amount of trade that's happening in this system. But if fertilization is prolonged or severe, this could actually lead to the evolution of reduced mutualist cooperation. And we have empirical evidence that that can happen. So this is a study from Kellogg Biological Station that showed that 20 years of annual nitrogen fertilization led to rhizobium populations specifically and whole soil microbial communities more generally that were less beneficial to plant growth. So we do have evidence that these predictions hold up in the real world. However, the effect of nitrogen on trade dynamics between plants and rhizobia is not the only mechanism that could cause this effect. And that's because nitrogen has lots of effects on plant ecosystems besides this impact on trade. So first of all, nitrogen tends to reduce the species richness of plant communities, and it's especially harmful to legumes. Legumes do really well in low nitrogen soil because they have this in-house source of nitrogen. But if soil nitrogen is more abundant, the legumes generally can't grow big and fast enough to outcompete other species. And so in these long-term nitrogen addition experiments, legumes are greatly reduced or even gone entirely. Secondly, the more intuitively and visually obvious effect is that nitrogen makes plants get bigger. So this is a photo of one of the nitrogen addition plots at KBS, and you really don't need those corner posts to see where it's been fertilized. It's very obvious that those plants are much bigger. But the effect of this increased overstory biomass means that plants in the understory get less light. That light is all blocked from that big overstory. And a lot of legumes are these smaller understory plants. So legumes growing in nitrogen addition plots are also more carbon limited because they can't get enough sunlight. So if we return to this market diagram, and now we reduce the light availability of these plants, that's now going to shift our x-axis intercepts. And you can see that now, again, the benefit of trading with rhizobia has been reduced. Because it's harder for plants to acquire carbon, it is a bit more precious and harder to give away to that rhizobia. So again, it's still beneficial, but less beneficial than it was before. So now I've demonstrated that both an increase in soil nitrogen or a decrease in carbon availability both reduce the benefit of participating in mutualism. However, in the field, these aren't happening separately, they're happening simultaneously. So if I combine those two effects in this diagram, you can see that now the benefit of trading for rhizobia is really, really small. This is hardly a mutualism at all. There's still a benefit, but it's so small, it might not be a significant selective pressure in the field anymore. So all of these things could be contributing to that effect that we saw in the field of reduced mutualist quality. So for this project, I really wanted to dive into this why of rhizobium evolution and figure out which of these factors were more important for reducing the mutualist quality of rhizobia. Because theory supports these three different possible causes. So first, increased soil nitrogen could directly cause trade with rhizobia to be less profitable for plants. Secondly, decreased light availability, decreased carbon availability could indirectly cause trade to be less profitable. And finally, a decline in legume abundance means that even if trade dynamics between individual plant hosts and the rhizobia haven't changed, because those hosts just aren't there, there's less trade happening in the system overall. And all of these three effects means that rhizobia are now spending more time in the soil than they are in symbiosis with plants relative to an undisturbed system. And so that reduces the plant selection for quality mutualists. And this is predicted to cause rhizobia to become worse mutualists. And this could happen if there's selective trade-offs between traits that make rhizobia a good competitor in the soil versus traits that might make rhizobia a good mutualist. But even if these trade-offs don't exist, this could still happen through drift. So for this project, I set out to investigate both the repeatability and the reversibility of the evolution of reduced mutualist cooperation. And I'm going to talk about both parts of this project today. But first, I want to give a caveat that when I say evolution here, I'm using this in a very broad sense. I don't just mean microevolutionary changes in allele frequencies in rhizobium populations. I'm also talking about broader microbial community shifts. So my first question is, what ecological factors drive the evolution of reduced mutualist cooperation in high nitrogen environments? And to investigate this question, I experimentally evolved soil microbial communities in greenhouse mesocosms. I factorially manipulated nitrogen fertilization, light availability, and legume host density. And I inoculated all of these mesocosms with whole soil microbial communities that I collected from Kellogg Biological Station. And I really wanted this to be a real soil microbial community. I was worried that if I used a synthetic microbial community, I might miss out on any of those microbe-microbe competitive interactions that might be really important in the soil. So I paid the price of not knowing exactly what's going on in my system by not being able to track individual microbial taxa, but I have a more ecologically realistic soil community. I planted each of these mesocosms with a small plant community that had 4 grass species, 3 forbs, and 1 legume, and I picked Trifolium hybridum, which is a species of clover. I varied the proportion of clovers in the mesocosm. I kept the total number of plants in each mesocosm constant, but either had 1 or 8 clover plants to vary host availability. And then I replicated each of these treatment combinations 10 times for 80 mesocosms for this question. And I grew these mesocosms in the greenhouse for 5 6-week simulated growing seasons. At the end of the growing season, I'd harvest all of the biomass at the soil level and then let the pot sit fallow for 3 weeks, which would force the root nodules to senesce and the rhizobia to return to the soil. Then I removed all remaining root biomass, replaced half the potting media to reduce nutrient drawdown, replanted all plants from seed, and then waited another 6 weeks. And you can see from these photos that my ecological manipulations definitely had an effect. So these 2 photos on the right were taken on the same day, but the one on the top is low light and the bottom is low light. And those shaded carbon limited plants are definitely not doing so well. And then on the bottom, there's a really clear color difference between the fertilized and unfertilized pods. So that was a good first step. My manipulations are definitely affecting these plant communities. All right, so after just over a year in the greenhouse of these microbes evolving in these different conditions, I took a small soil sample from each musicosm and inoculated it onto individual clover plants growing in a common greenhouse environment to evaluate the overall plant phenotypic effects of these different microbial communities. And these were surprisingly striking. I saw a huge amount of variation in the size of these different clovers, where the only difference was the different microbial community treatments. So before I show you what my actual data looked like, I want to orient you to the format of this figure and then also go over what my theoretical predictions would look like on this figure. So first, the x-axis is going to be whether the microbes that these plants were inoculated with came from unfertilized musicosms on the left or fertilized musicosms on the right. The y-axis is total biomass for plant fitness. Color is going to indicate whether microbes came from unshaded musicosms in yellow or shaded in purple. And finally, pattern will indicate whether microbes came from low or high clover density musicosms. High is solid and low is striped. So first, I predicted that nitrogen fertilization would cause microbial communities to be less beneficial to plants. And if this is true, we'll see that the bars on the right are smaller than the bars on the left. I also predicted that a decrease in light availability would also make microbes less beneficial to plants. And here the purple bars would be smaller than the yellow ones. And finally, I also predicted that a decrease in clover availability would also make microbes less helpful for plants. And here the striped bars would be smaller than the solid ones. So this is the overall shape of the data that I would expect if all of these factors that I manipulated decrease mutualist quality in an additive way. But that's not quite what I found. So there's definitely interactive effects here, but there were effects of all the things that I manipulated. And with a three-way interaction, I know this is a complicated figure, but I'm going to walk through it one step at a time. So first, I want to point out that my one-year greenhouse experiment recapitulated the results of a 2-decade field experiment. And you can see that by comparing these two bars on the ends. So that yellow bar on the left is microbes from the treatment that most closely mimics the conditions in the ambient field plots. So that's low nitrogen, high light, high clover availability. And then this striped purple bar on the right is microbes from the treatment that most closely mimics conditions in the nitrogen addition field plots. High nitrogen, low light, and low clover. And the microbes in this treatment that received all three treatments, their mutualist quality declined so much that they were no longer mutualists at all. The plants inoculated with these microbes did no better than plants that were given no microbes at all. So that was a pretty dramatic result. I was pretty excited to see that. However, nitrogen on its own had no effect. So there was no difference in the plant fitness when the only difference in the treatments was nitrogen fertilization. So nitrogen alone, insufficient. Light might have had a small effect. There's no significant pairwise difference between these two treatment groups, but it's not a big p-value and the error bars don't quite overlap, so it's hard to say with confidence that light had no effect, but not a strong significant effect. Finally, just reducing clover host density also didn't have an impact. So none of these three factors were sufficient on their own to significantly degrade mutualist quality, but any combination of two factors was. So these factors are interacting with each other to make microbes less beneficial for plants. So overall, I found that an increase in soil nitrogen, a decrease in light availability, or a decrease in clover host density all contributed to a decrease in microbial mutualism, but these effects depended on each other. And this implies that in the field, nitrogen's effects on microbial evolution are largely mediated through broader changes in the plant community. And again, I want to stress that the only difference in the plants measured for this figure is the experimental evolutionary history of the microbes they were inoculated with. These plants were all grown in a common greenhouse environment, and all the microbial communities were identical at the start of the experiment. However, I was also interested in what would happen if I started with microbial communities that were already differentiated, and that brings me to my second question. Specifically, I wanted to know if mutualist quality would recover if nitrogen addition ceased. So for this question, alongside my original 80 mesocosms, I grew an additional 20 mesocosms that I inoculated with microbes I collected from the nitrogen addition plots at KBS, which have now been annually fertilized for over 30 years. I would have loved to do the full factorial manipulations on this set too, but for feasibility reasons, I couldn't, largely because KBS wasn't comfortable with me collecting that volume of soil from those plots because they're really small plots. So all of these mesocosms got highlight and low clover. And that's because that most closely mimics what conditions would be like in these plots if you stopped fertilizing. Light availability would really quickly return, but it would take a while for clover to recolonize those plots. But half these mesocosms were fertilized and half were not. And this figure is a really similar format to the previous one I showed you. The axes are the same, but now color is going to indicate whether that initial microbial community was collected from an ambient field plot in blue or a nitrogen addition field plot in red. So here I again predicted that nitrogen fertilization would make microbes less beneficial if they were collected from the ambient plots. But I predicted that if microbes were collected from the nitrogen addition plots, growing in unfertilized mesocosms would make them more helpful to plants than they were before. So overall, what I predicted is that over the course of the experiment, these microbial communities would converge in their plant phenotypic effects, such that the greenhouse fertilization treatment would be important, but the field nitrogen treatment would not be. But once again, not quite what I found. I did find that nitrogen made microbes less beneficial to plants. And this may appear to be at odds with what I just showed you, that nitrogen on its own was not sufficient. But recall that all of these plants had low clover availability. So this isn't the effect of nitrogen on its own. This is the effect of nitrogen fertilization in the context of low host availability. So here I did get the results that I predicted. But I didn't find any recovery of mutualism benefit. There's no difference between the two fertilization treatments with microbes from the nitrogen addition plots. So together, these results imply that it's easier to lose mutualist quality than it is to regain it. And I think there's a lot of different possible explanations for this. I think the most obvious one is just insufficient host selection. Because there's only one clover plant in each of these pots, even if that plant was trying really hard to reward good rhizobia, it just might not have been enough. So I think that's the most likely one. I don't think it's loss of genetic variation for mutualist quality, because I did see lots of variation in these different treatments, but that's a possibility too. There might not be a lot of variation for selection to act on. It could be a simple reduction in rhizobium abundance. Because the plants aren't there to reward rhizobia, they tend to decline in abundance quite a bit in these nitrogen addition plots. It also could be due to the way that symbiosis genes occur in rhizobia. So all of the genes for symbiosis and nitrogen fixation in this type of rhizobia are carried on plasmids. So those are really easy to lose if they're not selected for. But if those plasmids are lost from a population, it could be really hard to regain mutualist quality. And I do have a collaborator, Sierra Bedwell, at Illinois, who's helping me look into some of these possibilities, but unfortunately we don't have any answers quite yet. So for this first project, I showed you that increased nitrogen fertilization, decreased light availability, and decreased host density all contributed to a rapid decline in microbial mutualist quality. But these microbial communities were not able to recover mutualist benefits when they were grown in low nitrogen. So in this first part, I showed you how microbes changed over time when I manipulated the resource environment of plant hosts. And next, I'm going to talk about a project investigating how the legume-rhizobium relationship changes when plants have multiple symbiont options for investing their carbon. Specifically, I asked if multiple microbial mutualists create synergistic or antagonistic effects on plant hosts or on each other. So again, I'm sticking with the legume-rhizobium system here, but now I'm adding a third player, arbuscular mycorrhizal fungi, or AMF. So AMF are fungi that form these structures called arbuscules in plant roots, and then also these long strands of hyphae out into the soil. And mycorrhizae are really good at breaking down phosphorus compounds into forms that plants can use, and the high surface area of hyphae means that they're really efficient at foraging through a large volume of soil and getting nutrients and water for plants. And in exchange, the plants also reward the AMF with carbon. And this is what we call a tripartite mutualism or a multi-mutualistic system. And adding this third player makes this system so much more complicated than just a pairwise partnership. And theoretical predictions for this system in particular are mixed. And so that's why we wanted to investigate it. So with this project, we set up 4 different treatments. One where plants were given no microbes, one with rhizobia only, one with AMF only, and then a co-inoculation treatment where plants got both rhizobia and AMF. And since we have these mixed theoretical predictions, we decided to look at multiple different legume species to see if we get consistent results across species or if different species act differently. So we chose these 12 different species of prairie legumes that are native to Michigan. And because I'm looking at different species and I'm looking for interactive effects, I'm going to be displaying my results with log response ratios because that allows me to compare effect sizes across these different plant species that are much different sizes. And also because it makes it a lot easier to see whether you get non-additive effects of multimutualism. So instead of trying to compare that co-inoculation treatment to the additive expectation, you can just look at whether the response ratio is positive or negative. So the reason that we might expect non-additive effects in this system is because this third partner creates the possibility of indirect effects. For example, this AMF is providing the plant with needed phosphorus, which might increase the plant's accumulation of carbon. And that could increase its rewards to rhizobia, which could then increase its nitrogen fixation, increasing again the plant's ability to get carbon, and then increasing its rewards to the AMF. So here, the AMF is benefiting indirectly from the presence of the rhizobia mediated by the plant host. And this is what theory predicts will happen because these two symbionts are providing different rewards to the host. Theory says if there's different rewards, this could create synergistic effects, and the host will get more benefit from the combination of these two symbionts than we'd predict from their individual effects. And in this case, this synergism would be shown by a positive purple bar there. Conversely, theory also predicts that since both these symbionts are being rewarded by the same resource, carbon, this could create antagonistic effects because they're competing for a common reward pool. And if we see that, then our purple bar would be negative here. Also, these two effects could just cancel each other out, or there could be no indirect effects at all, in which case we would see no non-additive effect of multimutualism, and we'd have no purple bar. So these are the different possibilities for effects on plant fitness. But for how each symbiont responds, it's a little more complicated. I'm going to be presenting 2 different types of fitness proxies, an absolute fitness proxy and a relative fitness proxy. And this complication stems from the way that we measure fitness in these 2 different symbionts and trying to make fair comparisons between them. So for rhizobia, we count all the root nodules that the plant produces. But for AMF, since you have to score this under a microscope, you just take a set root segment. It's obviously completely impossible to look at every bit of root. And then you quantify the amount of that root that has been colonized by AMF. So we end up with percent root colonization. But these 2 different types of fitness proxies are telling us different things fundamentally. And so instead of comparing apples to oranges, I wanted to be able to look at fitness proxies that made sense with each other. And to demonstrate why that matters, let's imagine we have 2 different plants. So we have a really small plant that is heavily colonized by AMF, which is indicated by the darker orange bits on the root. And then we have this big plant that's only sparsely colonized. In which plant does AMF have higher fitness? Well, if we look at absolute fitness, it's higher in the big plant because there's a larger total volume of AMF in that root ball. However, if we only looked at relative fitness, if we only looked at percent colonization, it would be much higher in the small plant because a higher percentage of those roots are colonized. And so our decision on what fitness proxy to look at can really affect how we interpret our results. And so because of that, I decided to convert both of these fitness proxies into the other type and then look at all 4. So for rhizobia, to get a relative fitness proxy, I divided the nodule number by root biomass, and then to get an absolute fitness proxy for AMF, I multiplied percent root colonization by root biomass. So this is the general format of data that I'll be showing you on the next couple of slides. For the plant, we have the effects of the individual symbionts, and then any non-additive effects of multimutualism. And then for each of the symbionts, there's only one effect, the effect of one symbiont on the other. There's no effect of multimutualism because you can only compare the co-inoculation treatment to the single inoculation treatment. Symbiont fitness in the uninoculated treatment is definitionally 0. So there's only one effect, but then there's these two different types of fitness proxies. All right, so from my actual data, this is what we found looking across all 12 species. First, we found that AMF here weren't actually mutualistic in this project. There was no difference in plant size between the uninoculated and the AMF treatments. Rhizobia, though, were good mutualists. Plants were quite a bit bigger, but there was no clear effect of multimutualism. There's a slightly positive effect size, but it's not significant, so we can't say with confidence that there was no effect or that there was a synergistic effect. Similarly, there was no clear effect of AMF on nodule number, but AMF did appear to increase nodule density. Again, this is not statistically significant, but that's a big effect size and really big error bars. So what this is telling me is that it's probably not that plants aren't responding. There's probably just a lot of variation here. And we'll get back to that in just a second. The more interesting thing here, though, I think, is that the effect of rhizobia on AMF really depended on which type of fitness proxy you looked at. So rhizobia increased absolute AMF fitness, but decreased percent colonization. And this is due to exactly that effect that I showed a couple slides ago, because rhizobia are making plants bigger. And so here, even though rhizobia are decreasing the relative investment of plants in AMF, AMF as a whole are still benefiting from the presence of the rhizobia because plants are so much bigger. So this is what we found across all 12 species, but was it consistent? I already alluded to this, but no, it's not even a little bit. And in fact, if you look at these overall effects, which I just showed you and are highlighted now on the left, you'll see that there was not a single species that responded in the same way as the overall across species effects. So I'm not going to say I was completely shocked by this because it's ecology. Things are always going to be weirder than you expect. But yeah, there's just so much variation here. If you look at that middle panel, you'll see where those huge error bars came from. I mean, it's like there's different directions, different magnitudes. Every species is just going their own way. And this did make us glad, though, that we decided to look at so many species because we would have come to vastly different conclusions if we just picked one or two. For example, if we looked only at Astragalus, we would have said, wow, there's this really strong effect of synergistic multimutualism. Plants do way better with both symbionts than we would have predicted based on the two on their own. But if we, opposite, Astragalus is antagonistic, Tephrosia is synergistic. So these two species are having a completely opposite multimutualistic response. And about the only consistent thing we found is that rhizobia tend to be mutualistic. Almost every species has a strongly positive response to rhizobia, but even that's not universal because these two species, Lesbidusa capitata and Desmodium paniculatum, both did not respond to rhizobia. So there isn't a single thing that all of the species are doing consistently. So what do we do with this? I'm still working on that. If any of you have thoughts, I would really love to hear them. Because right now I'm just like, well, there's variation. That's interesting. There is practical implications of these results, though. As I mentioned, these are all native prairie species. And prairies are really important targets of restoration because we have destroyed pretty much all of our native prairies. And people are trying to change that, which is great. But late successional legumes in particular are really difficult to get established in prairie restorations. And because legumes are these nitrogen fixers, they can be really important components of ecosystems. So we really want to get them going in these prairies if we can. But these results indicate that you can't use a one-size-fits-all approach if you're trying to make sure these legumes are doing well. And that's not what land managers like to hear because that adds time and expense and difficulty. But this could also potentially help answer some questions if people are having trouble with these legumes. So I wish I had like a nice jazzy like summary sentence for this slide, but I really don't. Like I said, I would love to discuss these results more because yeah, I'm a little bit at a loss. But I think it's cool that all of these species are doing something a little bit different. And it really does emphasize that when you have these more complicated systems, these tripartite systems rather than pairwise systems, it can be really difficult to predict what's going to happen. Because even within the same plant family, you'll get a million different responses. So that's all I have. To wrap up, I first showed you that nitrogen made microbes less mutualistic, but the effects of nitrogen were largely indirect and mediated through other changes in the plant communities. And then I also showed you that multimutual effects are hugely variable in magnitude and direction, and that root symbionts have complicated effects on each other. And these symbiont effects can really shift in direction depending on what type of fitness proxy you look at. So that's really important to consider when you're setting up experiments. So with that, I will thank my funding and everyone who helped out with all these projects, and I'd love to take any questions.
For your second experiment about these differences across the species, is there any phylogenetical sign of these effects? Or?
I've been looking at that, and not really. So there is a small phylogenetic signal in the effect of AMF on shoot biomass, and strong phylogenetic signal in the effect of rhizobia on root-shoot ratio, but nothing else. I was kind of surprised about this because like we've got these pairs of congeners and then we've got 3 desmodiums, but doesn't seem to be a strong phylogenetic signal, no.
Yeah, I was curious for your second experiment, like where did you get your AMF and your rhizobium from?
Yeah, that's a good question. So we used 6 different strains for each species, 6 AMF and 6 rhizobia, and these were all isolated from native prairies where these plants are growing. So with a mix of 6 different strains, hopefully there's at least one there that they get along well with. We didn't just have a common inoculant for all 12 species, we really tailored it to that species. Yeah. Yes.
Did you look at any of the micronutrient concentrations in the soil, particularly like the ones that impact the nitrogenase enzyme?
I haven't. So I've just had the data for this project. I keep saying we set this up, but like it wasn't me. I just got the data. I've only been in this position for a year. But it was a really low nutrient potting media mix in the greenhouse. So I would think that there wouldn't be a lot of nitrogen around, which is probably why we saw that rhizobia did tend to be helpful. But yeah, I don't have a good answer for you on that.
Yeah, I'd be excited to learn more.
Yes.
I was curious for the first chapter with the total biomass metric, if it differed at all in like the root to shoot effects.
Let me see if I have a slide for that. I did at one point. No, that doesn't have root to shoot. I have that data somewhere. I don't remember if I had a strong effect or not. I don't think there was anything big. You get the expected response where having any helpful microbes makes root to shoot lower, but I don't think there were major differences between treatments. I think there were some, but nothing huge, if I remember right.
Very cool.
Yeah, thanks for the talk. I was curious, like, so in those models that kind of take this trait into consideration, and often in these models we kind of think about okay, there's this kind of exchange going on and we focus on those resources, but, and I guess it kind of connects to the question about micronutrients. They're at the same time still competing for everything else.
Yeah.
And I don't know if there's any explicit way that's kind of brought into account, and if there's a way that that can maybe explain sometimes some of these different outcomes, that it's based on a variety of other traits that are there that changes sort of competitive hierarchies between these organisms, you know, that, you know, yes, we can say they have a symbiosis, but that's really just for that particular exchange. And overall, you might see fitness benefits, but there's so much underlying that I have a hard time grasping sometimes, but I don't know.
Do you mean in the plant communities or the microbial community?
Even just directly with what we consider the symbiont and its host species, right? It's, yes, there is this mutualism going on, but actually it's the balance of a mutualistic interaction for particular, in an exchange between certain resources, but then there is still competition for everything else. So depending on how much availability there is of everything else, that might change the outcomes. And I don't know, I haven't wrapped my head around that myself completely, but like if that's in any of the modeling kind of explicitly brought into account, or that is sort of made in abstraction sort of with these isoclines that kind of take it all into account, but it's still conditional, right? So it's going to, if your conditions are different, those isoclines are going to look different. And I don't know. I don't really know what fully the questions is, but I wonder if that kind of competition for everything else can be a potential explanation.
Yeah, I'll have to keep that in mind.
Thanks for the talk. That was super impressive amount of work. It's really cool. I'm curious about the trends that I'm sort of just maybe eyeballing and not really, I don't know if this is right at all, but it seems like the AMF in the cases where it's negative to the plant are all the cases where the mutualistic effect of the combo seems high. Is there any reason why that might be a reasonable biological expectation, or is there something?
I think what's happening there is not that plants are doing better in the co-inoculation treatment than they are with the rhizobia treatment, but that if you average a negative effect of AMF and a positive effect of rhizobia, it would be 0. But then in the co-inoculation treatment, they're doing about as well as with just rhizobia. And so it looks like a really big effect of multimutualism because it's bigger than the additive expectation. But it's not that they're doing extra well, it's that they're just still benefiting from the rhizobia and not really doing much with the AMF.
In another question. Have you seen how, what is the frequency about the investment in these interactions in natural populations? Because I'm wondering if for one species, they invest more in rhizobia, but not in the other one. So maybe testing, like, is expected that the effect will be different.
It probably would be different. I don't have an answer for you. I haven't studied these in the field at all. It's really hard in the field because you have to like dig up the whole root ball somehow and then disentangle it from all the other roots and figure out, you know, what plant you're looking at. I do know that nodulation tends to be lower in the field than in the greenhouse. But again, it really is just hard to study. And that could be due to, you know, plant competition or insufficient light or, you know, any number of things. I don't know as much about AMF. I'm really new to the AMF world. So I don't have an answer for even like, does AMF colonization tend to be lower or higher in the field? I assume it's different. It probably depends a lot of things on like, I mean, this environment is so artificial, right? Because not just is it really short compared to all these, you know, perennial plants. There's only one annual in this group, but they're really small pots. And if the benefit of AMF is foraging in a big volume of things, then like maybe it's not surprising that we don't see that benefit in these tiny little pots. Yeah, would love to do it in the field, but if I had 2 more years of postdoc, I would do that.
Any more questions? All right. Thank you, Mackenzie.
Thank you so much.